Welcome back, students. This is Module 2.2.5, Active Processes. You'll recall that we have just finished studying passive processes, which require no energy, but while that sounds very good, there is a serious limitation to passive processes, and that is they are limited to moving substrates or solutes down the gradient from an area of higher concentration to lower concentration. If you need to move things in the opposite direction from lower concentration to higher concentration, then you must have an input of energy, and that's when active processes come in. So active processes, we have substances that are moving from an area of lesser concentration to an area of greater concentration up the gradient. This may be likened to going upstream in a river against the current, against the flow, and it requires an input of energy. Typically in cells, the energy source is the molecule ATP. This is actually a nucleic acid, and we'll learn more about it when we study cellular respiration. There are two types of active processes that we're going to study. One we simply call active transport, and the other is vesicular transport, transport in little membrane-bound vesicles, also called bulk transport. First, we're going to consider active transport. In active transport, we have integral membrane proteins that will push substances across the membrane up the gradient. Integral membrane proteins simply means that the proteins are embedded in the membranes. And again, these protein carriers are selective, and they are, of course, powered by ATP. That's one of the things that separates this process from another process that uses selective integral protein carriers. That was facilitated diffusion. It also uses those specific carriers, but they were powered by the concentration gradient and move stuff from higher to lower concentration. Active transport moves things in the opposite direction from lower to higher and requires an input of energy since the gradient won't effectively move it. Let's look at an example. This is a little bit complicated, but if you look at it carefully and understand, I think you can benefit from it. So start in the top center here, where it just kind of shows us the calcium pump. This is a protein, and it is integral to the membrane, and it's showing calcium on the outside, the little black dots. They're more on the outside than the inside. And yet, if we want to take calcium from inside the cell and move it out in such a circumstance, that would be moving from lower concentration to higher concentration, and that's where the calcium pump comes in. In fact, we can see this fuel molecule, ATP, come over to the pump, and it binds to the calcium pump. That changes the conformation or the shape, and it allows calcium to enter the pump from the outside, from the inside, and it binds inside. The ATP then transfers a phosphate group, it transfers energy with that phosphate group, and this causes a shape change, and it causes the calcium to be released on the outside, and so it's gone against the gradient and moved out. And as the calcium leaves, the shape of the pump goes back to what it was at the start, and it's ready to do it all again. So this kind of shows the steps that are involved in that process. Here's a little animation that shows the same thing. Active transport requires energy. You can see the little star of energy, that's the ATP, and it energizes that protein. And so ATP is that energy, and it moves against the concentration gradient. And the second process that we're going to consider after just active transport, which active transport is very common, is vesicular transport or bulk transport. And this is movement of a large amount of substances into or out of a cell inside a membrane. And a little membrane full of stuff being transported is called a vesicle, hence the name vesicular transport. There are two types of vesicular transport or bulk transport, exocytosis and endocytosis. Exocytosis, you're moving substances out. The etymology of the word, exo means out, cyto means cell, endo means in, cyto still means cell, so moving substances into the cell, moving substances out of the cell. Turns out that there are two types of endocytosis, two ways that we move things in, or we classify two different types of endocytosis. phagocytosis and pinocytosis, and we'll talk about each of those in turn. Let's make sure that we understand what vesicular transport is first, though. And so this actually shows the process of a vesicle being formed. It's an intracellular, which means inside the cell, vesicle. And this shows a similar process in a still below the animation. It shows substances outside, and it shows the cell membrane pinching and forming this little vesicle, which pinches off and moves inside the cell. All this movement of the membrane is powered by ATP, so it is indeed active transport. Excuse me. Now that we know the basics of vesicular transport, let's look at the two types. The first being exocytosis. Exocytosis, in that process, we have vesicles are formed inside the cell, shown by the first arrow, and these vesicles then fuse with the plasma membrane and release the substance outside the cell. And this is powered by ATP. And so exocytosis, moving substances outside the cell. Here we can see a little animation showing the same action. So the vesicle comes over, fuses, and the contents of the vesicle is released to the outside of the cell. So that's exocytosis. Many cells that secrete things, some glandular cells, work in this way. Now let's look at the other type of vesicular bulk transport, which is endocytosis. And remember, we said there are two types of endocytosis, two classifications of moving things into the cell in bulk. The first of those is called phagocytosis or phagocytosis. You can say the G soft or hard. I've heard it both ways. Just say it with confidence and a knowing smile. In phagocytosis, solid particles are ingested into the cell. So we can see this little green rectangles of food, and there it's ingested by pseudopodia, or false feet, is what they sometimes call these little projections of the cell membrane that surround a food particle. And then a food vesicle is formed inside. And so that's phagocytosis. In pinocytosis, instead of the substance that's being moved inside the cell in bulk being solid, it's liquid. And so that's sometimes phagocytosis is called cell eating, sometimes pinocytosis is called cell drinking. So here we go. Here's phagocytosis. And projections of the cell called pseudopods or pseudopodia surround the particle and engulf it. For example, white blood cells will engulf invading pathogens or bacteria, or an amoeba can engulf a food particle for a meal or a food organism, again powered by ATP. Here is a photomicrograph of a blood cell engulfing a invading cell, a parasite. And notice it doesn't look like the classical example, but it just shows you that we give you kind of a typical example, but reality may be somewhat different, so be aware of this. A second expression of endocytosis is pinocytosis, and this is known as cell drinking. And really the only reason that we mention it here is because of its great importance in the transport of nutrients from the intestine inside the intestine cells. And so this is how many of the nutrients in vertebrate animals get into the body, is by the process of pinocytosis. And so remember, pinocytosis is defined as the engulfment of liquids with solutes by unfolding of the cell membrane. And it too is powered by ATP. And as I've already just stated, it's very important in absorptive cells of the intestines. And that will conclude this module, and we'll be back soon.